So the history of trials started back in biblical times with Nebuchadnezzar and Daniel and a diet observational study. We've come through the 1500s with advances in anatomy, the 1700s and 1800s with the first recorded comparative studies, with the first placebos being recorded. And this has all set the stage for what is to come in the 1900s and now. So in the early 1900s, there were randomized controlled clinical trials, and blinding became part of the gold standard for trials. Now we start to get into some of the heavy hitters of statistics. So for any of our biostat students, these are names that you've seen in your courses. For those who've had introductory biostat, certainly Fisher's exact test you have heard, but the rest of these statisticians may not be as well known to you. So we have R.A. Fisher here on the left, 1890 to 1962. We have Jerzy Neyman, 94 to 1981. Carl Pearson, very famous statistician, and his son, Ergon Pearson, and then Frank Yates from 1902 all the way to 1994. So we're starting to get into some contemporary statisticians here in the early 1900s to late 1900s with the development of controlled trials and blinding. It was in the 1920s that the theory of randomization emerged, and this was 1923, R.A. Fisher, and he is largely credited with codifying the concept of randomization in this paper. Fisher and McKenzie from 1923, Studies in Crop Variation, and this is from the Journal of Agricultural Science. So actually a number of well-known statistical methods developed from farming or crops or forestry, and then also from animal husbandry. Crops and animals were a little bit easier to control rather than people. As we know, people like to do their own things, and that can be problematic when developing new scientific methods. And so the concept of randomization actually stemmed from crop variation. And they tied randomization at this point to an analysis of variance. So Anova is something that any introductory biostatistics course will talk about. It's the comparison of the variation within a group to the variation between groups. So are the means in these two groups sufficiently far apart to declare that they're different? And Fisher tied randomization to Anova, stating further, if all the plots are undifferentiated, and so what he means there is the homogeneity. So if the plot of land is fairly similar, so you took a plot of land that was divided, say, into 4 parts, and that plot gets sufficient amount of sun everywhere and the same amount of water, so it's making a homogeneous study area. If all the plots are undifferentiated, as if the numbers had been mixed up and written down in random order, the average value of each of the 2 parts is proportional to the number of degrees of freedom in the variation of which it is compared. So this is a very statistical way to say the average value of each of the 2 parts. So if you took the mean of the 2 sides of the plot, so if you split it into 2, if you took the mean yield overall, it will be proportional to the number of the degrees of freedom and the variation of which it's compared. So the overall mean is going to be relative to the number of means that are contributing to it. And this is basically the sense of the analysis of variance. We have an overall mean yield. So let's say we're growing corn. We have an overall yield of corn ears. And if we take a plot divided into 4 parts and we look at the mean from each of the parts, we want to know if each of A, B, C, or D plot produces sufficiently more or less than the overall mean. And that's where the idea of randomization came about, that you could randomly apply, say, a fertilizer to one area, more water to one area, nothing to another area, and more water and more fertilizer to the third area. That's actually a factorial design. In 1923 and 1925, Neyman also arrived at a similar conclusion about randomization, but publication was delayed by Pearson. So Neyman-Pearson is a very famous statistical relationship. There are tests and methods that are the Neyman-Pearson method. And so it turns out that Fisher was able to scoop them. So two different bodies of thought had similar ideas, but a publication delay gives Fisher the credit. In addition, there's some controversy erupted in 1927 when this paper was actually published by Neyman and Pearson, not crediting a Russian statistician, Alexander Alexandrovich Chuprov, who also came up with this idea and contributed, and they did not cite him. So really, it should be the Neyman-Pearson-Chuprov method, but it has stuck with Neyman-Pearson. As any number of these things happen over the course of history, there are many statistical controversies that have come to light since, and this is just one of them. At any rate, we now have the concept of randomization. The late 20s and early 30s in the 1900s were very active in terms of statistical theories and randomization. There were 3 simultaneous publications in 1935 that are basically the foundation of randomized experiments. So here's Fisher again with his Design of Experiments book. Neyman with the cooperation of 2 Russian statisticians that this time he did decide to collaborate and publish with in 1935. The Statistical Problems in Agricultural Experimentation, and this was published in the Royal Statistical Society Journal, and that's a very well-known statistical journal. And then Yates in 1935 also simultaneously published Complex Experimentation, which was a supplement in that same Royal Statistical Society. So a lot of publication on the design of experiments, and many of these designs are actually still used today, and they transferred from agriculture into human research. In 1943, we had the first double-blind study. So this was not a randomized trial as we think of today. It was the Medical Research Council Potulan Clinical Trials Committee. They enrolled 1,000 British participants with colds, which, of course, modern medicine is still trying to cure. But they decided to test an extract of penicillin. Both the patient and the physician were blinded. And there were 2 statisticians on the committee. So they knew the importance of statistical input way back in 1943, and they were not wrong. So this was not randomized. There was no randomization procedure. It was just allocated. Somebody said this person got this and this person got that, and it was given to the physician to give to the patient. And neither the patient nor the physician knew whether they were getting penicillin or our placebo, of course, which was introduced back in the 1800s. And that takes us to the late 1900s. And that gets us to the first randomized trial. And by the way, they did not cure the common cold. Of course, we still have that problem today. And people will continue to try to study. But I guess we just have to live with the sniffles. By 1946, Sir Austin Bradford Hill, who lived from 1897 to 1991, incidentally, the year I graduated from high school, Sir Austin Bradford Hill developed the first randomized trial. So the prior experiment with the penicillin was not randomized in the sense that we knew it today. If you are in epidemiology or if you've done a lot of causal research, then you know about Bradford Hill criteria about causation. So it lists the Hill criteria, and they've been amended since then, about how to draw causation from causal studies. As you know, correlation does not equal causation. The whole point of randomized clinical trials is so that we have a starting point for an intervention and you can see what happens before and after. In many large cohort studies, some of which are run out of the School of Public Health, there is no randomization. But there is a temporal component. So you can actually know prior to someone developing a disease, say stroke, that they have not yet had a stroke, and follow them until they do have a stroke and try to see if you can develop some causal relationships from that observational study. Correlation does not equal causation. I can't say that enough. If you are curious about how to develop or draw causal inferences from observational research, then go ahead and take a look at the Bradford Hill or Hill criteria for causation. And Hill had the MRC streptomycin and tuberculosis trial. So they were looking at streptomycin and TB. In 1947, the trial begins. So streptomycin is available, but it was limited. So ethically acceptable for control subjects to be untreated at the time. That is another thing that has changed. If there is a known treatment available, it is very difficult to get placebo-controlled studies approved in this day and age. Maybe for short periods of time if it's early onset and benign disease. But if there's an accepted treatment, we look at treatment-controlled trials rather than placebo-controlled. But 1947, it was ethically acceptable to not treat people for TB. So the statistician, here we go again, another statistician involved, Sir Austin Bradford Hill, devised the randomization, and that is our British spelling. We use a Z over here, over the standard every other person allocation. So the prior controlled trial just gave every other person the treatment. And the issue with that is, of course, is that if you're an adept investigator or physician, you may be able to tell what the real treatment was, especially if the effect was pronounced. And then from there on, you would know what every other person was getting. So randomization really helped with that. Determination of whether a patient would be treated by streptomycin and bed rest was called the S case. So they got streptomycin and bed rest, or by bed rest alone, called the C case. The details of the series were unknown to any of the investigators or to the co-coordinator, or to some extent the patients. Thankfully, we've dropped it to just coordinator now. And were contained in a set of sealed envelopes, each bearing on the outside only the name of the hospital and a number. So here we begin to see the blinded identification of participants. Austin Bradford Hill, or Sir Hill, had actually devised the treatment allocations, and basically only the pharmacist would know, but the treating physicians on the hospital ward did not know what people were getting, or the nurses or coordinators, because everyone was getting bed rest, and some people were getting strep, and some people were getting placebo. The trial results, 4 of the 55 S cases, so those receiving streptomycin and bed rest, so 4 of the 55, which are 7%, and 14 of the 52 bed rest only, 27%, died before the end of 6 months. The difference between the two series is statistically significant, and it's in fact easy to recreate this chi-square or Fisher's exact test. MRC Streptomycin and Tuberculosis Trials Committee was published in 1948 in the British Medical Journal, showing that the streptomycin was effective for treatment of TB. Hence, therefore, it would actually be unethical to not treat patients, to just give them bed rest. So that's brought us up to basically the late 1900s and early 2000s. What's next? Well, once this trial was initiated and reported on TB, randomized clinical trials started becoming the norm. There are a number of pivotal trials, and the ethics, of course, of the trials began to crop up. And that is what we'll discuss next. What are the lessons learned from the quote-unquote modern day clinical trials?